Hi again. Okay, remember in the last video we shared how the action potential coming from the axon ends up being able to be transferred or jump over that synaptic cleft based on the use of the neurotransmitter. And once that neurotransmitter binds to its receptor, it starts the action potential in the sarcolemma. So this action potential travels down the T-tubule and back up, and it keeps going. But the part we need to talk about now is what happens here in this T-tubule. So when this action potential travels down the T-tubule, it's going to send a message to this sarcoplasmic reticulum, this yellow organelle here, which is a modification of an organelle we have in all of the rest of our cells called the endoplasmic reticulum. And here in the muscle cells, its job is to store calcium. They're in green here in this figure. So when the action potential travels down the T-tubule, it triggers the release of calcium into the middle of the cell. If you can see up here in this top version, remember that the bulk of the muscle cell is filled with these myofibrils. And remember, the myofibrils are sarcomere, 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 right, all the way down. So what we're doing is we're flooding the sarcomeres with calcium. So calcium comes out of the sarcoplasmic reticulum, and it's now available to bind to each of these sarcomeres. Let's go on to the next slide here. All right, and we're going to use this figure to show what happens in the sarcomere once that calcium is released. So here at the top in this figure, let's see, where am I? Way up here. Here's the calcium that came out of the sarcoplasmic reticulum. Remember, that calcium binds on to the thin filament, and when it does that, it actually causes the thin filament to shift in a particular way that exposes the binding sites for the myosin heads on the thin filament. So as soon as that thin filament shifts, particularly the green line here, which is called the tropomyosin, this myosin head binds to that binding site on actin. As soon as it binds, it's going to pivot forward, and as it pivots, it's going to be what pulls the sarcomere towards the middle. Remember, we have the sarcomere here like this, and these heads go from this direction inwards, and it pulls the pulls the thin filaments on either side towards the middle of the sarcomere. So here in this figure you can see what we're talking about. And the myosin heads keep doing their job, and they ratchet inward, and that pulls the thin filaments in over the top of the thick filaments. Now, here is the motion again of the pivoting that happens. ATP then comes in and binds. Remember, ATP binds and it breaks that head off of the thin filament. The head also has a special job, remember, of being an ATPase. Do you remember that term? And that just means that the head can break down ATP. And what does it break ATP down into? It breaks, let me find my spot here, it breaks ATP into ADP plus phosphate plus energy. Sorry, my pen is all kinds of crazy. So ATP gets broken into ADP plus phosphate plus energy. And this energy is what we use to cock or to put that myosin head back into its high energy position again, down here at this bottom part of the figure. So ATP is used to recock that myosin head, and as long as calcium is still present, it's going to bind again to the myosin binding site, it's going to pivot again, and it's going to continue bringing that sarcomere in. So as long as calcium is present, then that, and ATP is present, the myosin head will reach up and attach, it will pivot, ATP binds to break it off, ATP gets broken down to recock the head, it grabs again, it pivots, ATP breaks it off, ATP is then used to recock the head, and the whole cycle continues over and over and over again. And that's how the muscle contraction actually occurs.